This is just a really quick CFU practice. Hopefully I've drilled this into your head so much it's going to be something you can do in your sleep by the time you get to the test, but maybe that's not true today. To quantify microbes in an air sample, what do we calculate? That's right, it's CFUs per square foot hour. To quantify microbes in a water sample, what do we calculate? CFUs per mil is how we're doing it in this class. That's right. So how many colony forming units do you approximately see on this plate? All right, good job. It is 25 is the closest number. I mean, you might have gotten a slightly different number. It's not a big deal. All right, so we have a plate that is 1/5 of a square foot. That's also 0.2 of a square foot exposed to indoor air for 30 minutes. What's the CFU per square foot hour? This is the same plate as before that you counted 25 CFUs on. So now you have the area, exposure time. What's the CFUs per square foot hour? All right, so it was 250. Let's find out why. There was 25 CFUs. We divided by the product of the area times time, 0.2 times 0.5, because 30 minutes is half an hour. Or we just intuitively know, since we only sampled 1/5 of the square foot, we have to multiply our CFU number of 25 by 5. And since we only exposed for half an hour, we have to multiply it by 2. So that's basically 25 times 5 times 2, or 25 times 10, 250. How many CFU do you see on this plate? All right, 17 is the best answer. And if this plate was spread with 0.1 or 1/10 of a milliliter using a 1 in 1,000 dilution, what's the CFUs per mil of the original water sample? All right. Good job, everyone. So you took your 17. You knew that we only sampled 0.1 mils, so we have to multiply by 10. And we diluted 1,000-fold, so we have to multiply by 1,000. The other way you could have done this is take 17 divided by the product of the volume times the dilution. So the volume is 0.1 mils and the dilution is 0.001. I had to do that in my head. And that's the same thing as multiplying basically by 10,000. All right, let's have a quick peek at who made the podium today, but I know that y'all were doing a great job, even if you didn't get on these very top spots here. So thanks for playing, and hopefully you feel a little warmed up this morning. All right, good job, everyone. Now, I'm also aware that it is not everyone's forte to operate under pressure. So I do that partly because that is a requirement, to operate under pressure on the exam and in your chosen field. So this is kind of ramping you up to that. All right, so our focus this week is microscopy, and we're going to look at today focusing on the principles of microscopy. If we have time, and we may well have time, we'll move into this material on staining. But if we don't finish all of it, which we probably won't, we'll finish that up on Wednesday. And that'll leave some time to have a longer discussion about the Unknown AD project and other things. Okie dokie. So one of the major problems in the field that we talked about in the very first day is that the stuff that we're trying to understand in this class is really small. We can't, by definition, can't see it with our eyes. One solution, simple one, is what tool can we use to kind of get around this problem? Right. You can use a microscope, right? A microscope is useful because it magnifies things. I would say it's more intricate than a magnifying glass, so it also provides, some kind of resolution. The other problem that I've talked about a little bit, but probably if you haven't been to class yet, or lab yet, and most of you have not, we'll talk about this a little bit more in lab, is that microbes are transparent. They're see-through. They don't have a lot of pigment, most of them. Some of them have a little bit, like those that are photosynthetic have some green pigment, but a lot of them are completely transparent. So what is one way to work with this if microbes are transparent? Right. We can color them, basically. It's not substantially different from just setting them in like food coloring dye. So we can dye them. So these are the things we're going to focus on this week. They're not the only tools in the arsenal of microbiologists, especially now with all of the newer technology, and genetic sequencing is a big tool in the toolkit of microbiologists, just like it is of any biologist nowadays. But we're going to get to that in genetics. So first things first, this is a review. What units are typically used to measure cells? Put that in the chat really quick. So we're just getting used to using these units and talking about them and feeling comfortable and familiar as if they were our good friends, meters and centimeters, but they are in fact micrometers. Micrometers are 1,000 times smaller than millimeters and 1,000,000 times smaller than actual meters. And viruses, of course, we use the other even smaller unit, 1,000 times smaller than that. We get into the nanometer range, exactly. Now, thinking about the size of things, right? Because if we're going to deal with, a tool that allows us to see small things, we might have to know how small the things are that we're trying to see. What is a typical size range of a eukaryotic cell? Right. In my curriculum, you'll see that it is usually described as between 10 and 100 micrometers. Now, there are exceptions to this. There are some yeasts that are smaller, and there are some protozoa, like amoebas, that are bigger, and there are also some algae that are bigger. But this is actually sort of a little bit human-centric. So the smallest human cells, red blood cells, are around this size, and bigger ones are around that size. What about prokaryotic cells? How big are they? Yeah. Typical size range is they can be skinny, maybe a half a micrometer, and they can be a little bit long, maybe up to 5. Around the 1 micrometer range is also a good way to remember it. When we look at things with our eyes, what's the smallest thing? A typical, really healthy eye, not mine. Now I can't see anything without reading glasses, but limit of resolution of the human eye is typically described as being what? That's correct. 200 micrometers, or what would that be in millimeters? What would that be in millimeters? Convert it to millimeters in the chat for me. Yeah, 0.2. Good. 0.2 millimeters or 200 micrometers. So can we see microbes with the naked eye? Right. The answer is no, not really. I mean, if you want to be a purist about it, then as I mentioned, there are some eukaryotic cells that are a smidgen bigger than this, right? And if they were just sufficiently big enough, like some amoebas, what we would see is a speck, but it's so small that for all intents and purposes, we can say no. I've mentioned this before, I'll mention it again, that I think it's really helpful in this field when we're dealing with things that are unable to be seen with our eyes to use the wonderful animation techniques of today to zoom into that tiny world to start to get comfortable with it, because then you can hold a picture in your mind. And for most people, that's really helpful. So this is a zooming in animation tool that's really great. Here is a comparison of different sizes that I've given you before. Viruses being like 1/10 of a micrometer, prokaryotic cells like bacteria being around 1 micrometer, and then a variety of sizes for the eukaryotic cells, from human red blood cells at 10 micrometers, slightly smaller than that, yeast at 5, amoeba was larger than that at 400, but around the 100, 10 to 100. So this is just an electron micrograph zooming in on the head of a pin. This is the head of a steel pin. Then as we zoom in, we can see that it's covered with bacteria that we would never be able to visualize with our eyeballs. Okay, the other challenge that I feel like people face is getting really comfortable with the units. Because micrometers and nanometers are smaller than we use in our daily lives, it can be a little bit hard to just have an intuitive number sense about them. And so we're just going to do some simple calculations just to kind of, again, warm up, get comfortable with them. And, a reminder, hopefully this is obvious now, but, these Zoom sessions are an opportunity for me to surface some of the places that I have found students need extra help, or places we can review, or things that are particularly interesting, or ways we can apply the information so we care about it, but they're not comprehensive. So you need to spend that time looking at the video lectures that are really the meat of the class. Nonetheless, what is the way to convert 1 millimeter into micrometers? What are you going to get? Put that into the chat. Correct. 1 millimeter is 1,000 micrometers. Good job. Okay, now into the chat, you're going to put this and then that. So 1 micrometer, which is what we use to measure cells, is either what in millimeters or what in nanometers. So do those conversions. Good. Yeah, just getting, we're doing some simple stretches at the gym, nothing strenuous. Hopefully no one pulls a tendon doing this or whatever. Okay, yeah. So when we want to go from micrometers, which are small, to millimeters, which are bigger, we divide by 1,000, so we get 0.001 millimeters. We're going from micrometers to nanometers, Nanometers are smaller, there's going to be more of them, so we multiply by 1,000 to get 1,000 nanometers. And 1 nanometer is how many micrometers? Right, so we have a small unit, we're going to a bigger one, we're going to have to divide to go to that bigger one, divide by 1,000, right? We get 0.001 exactly. Great. So now let's do it with numbers that are not 1. 0.2 micrometers is how many nanometers? Great. Awesome. And if you end up getting it wrong, just know I'm watching the chat. I'm not going to remember it was you. I just am watching to kind of get a sense of what the numbers are rolling in. So you don't have to feel like I'm watching you. Nobody else is watching you either, because all like looking at their own number. 0.2 micrometers, you want to go to nanometers. Nanometers are smaller. That means there's going to be more of them because they're smaller. So we have to multiply by 1,000. 1,000 nanometers per micrometer gives us 200 nanometers. 200 micrometers is how much in millimeters. 200 micrometers is how much in millimeters. Great. And you also want to be getting in the habit of making sure whenever you report a number, you're using units. Just be in that good habit. Again, I'm not going to notice if it was you or not you that left your units off. But being in that habit makes it possible not to drop some units on a test or someplace where it matters for grades. Good job. Okay, so we're going from micrometers to millimeters. Millimeters are larger. We're going to have to divide by 1,000, right? Or we could just put this in. We could say, well, I know 1 millimeter is 1,000 micrometers. So, oh dear, my cat just caused a ruckus. So we get 0.2. Now here's a picture. This amoeba, as I mentioned before, is about 400 micrometers in its long dimension. What is that in millimeters? I bet you didn't think you'd be doing math all Monday morning. It's not going to be all morning, I promise. We're going to get through the math really quick. Yes. So we're going from micrometers to millimeters. Millimeters are larger. We're going to have to divide by 1,000. We get 0.4. Yeast, this is a beautiful electron micrograph, a pseudocolored micrograph of budding yeast. This is the guy that you use if you're making bread. It's also used in the brewing of beer and wine. They're about 5 micrometers, and they're like on their waist, their equator, if you will. So what is that in nanometers? We're going to a smaller unit. There's going to be more of them. We have to multiply. 5 times 1,000 is 5,000. Now, first, we're going to look at this in nanometers. So, and just answer it in nanometers first. So coronavirus is how many nanometers, and how will you know? So I'm not telling you, right? I'm showing you a picture, and I'm giving you a scale bar. So here are the coronaviruses, just for funsies on a filter. But if we look at this zoomed in image, we can see the coronavirus at a higher magnification, and here's the scale bar on this magnification. And so what is the width of that coronavirus in nanometers? That's right. Width of that coronavirus in nanometers is about 100, right? And just so happens that the scale bar is the same size as the virus. Doesn't always work that way. We'll talk about how to do conversions from a scale bar, but this one's nice and easy. So what is that in micrometers? Great. So we're going from nanometers, a small unit, to micrometers, a larger one. There's going to be fewer of these, just like if you were measuring something in centimeters and convert it to meters, there's going to be fewer meters. So we have to divide. We have to divide by 1,000. And so we get 0.1. Now, if you're ever finding yourself confused, or you're just like under pressure, you're like, I know this, but it's the test, I'm kind of frazzled. Did I do the conversion? Because it's micrometers and nanometers, that's where you're going to get tripped up. But just sort of take a second to think, okay, well, actually, in this instance, micrometers are kind of like real meters, and nanometers in comparison are millimeters. You would never, if someone asked you, you know, this basketball player is like 2 meters tall, how many millimeters? You would not be like, is it 0.002 millimeters or is it 2,000? Like, that's not confusing, right? So just remember that if you're trying to go from a large to a small, or a small to a large, you could sort of transpose that into the real world, right? You could ask yourself, what if I was talking about millimeters and meters? Would I be confused then? Okay, another scale bar example. This one is harder. So we're going to do the width. So this is a rod-shaped bacterium. You can see E. coli is rod-shaped, so it has a skinny part, which is the width, and a long part, which is the length. We're going to do the width. You'll also notice that these rods are not all identically sized. Some are longer, some are shorter. So it's kind of up to you which one you measure. This is the scale bar. The scale bar is 5 micrometers from this side to that side. Do your best to estimate based on that how wide these E. coli's are. And I put it into the chat. And don't worry about being way off or being unsure how to do it. If you have no clue at all and you're just not comfortable volunteering a number, you can put a question mark. That's always allowed. But I'd rather you put a question mark than nothing, because it's not for me, it's for you. Keeps you awake. But what do you think the width is if this is the scale bar of these cells? And then we'll talk about it. Great. Okay. The waist, Lanisa. Yeah, we're looking at the width. Okay. So before I reveal my answer, I'd like to hear from someone who's quite confident in their answer and tell me how you figured this out. So go ahead and raise your hand if you feel comfortable telling us how you figured it out. I might ask just a couple people before we even talk about what the right answer is, so don't be afraid to, say something that other people maybe wouldn't understand or don't agree with. Erica Marston, go ahead.
At first I said it was like 2 but then you said it was the width, not the length. So I pretty much just took 5 and tried to see how many I could fit in there. And I didn't come up with the answer, but that's how I would try to find it.
That's how you were thinking about it. Great, thank you. All right, does anyone else want to share? That was a great answer. Does anyone else want to share how they came up with their number or how they were thinking about doing this? I mean, I see some great answers in the chat, so there must be a reason that you chose the number you did. I am going to cold call a few of you, and remember the rules of the cold call, so don't freak out. If you don't want to tell me, you can just say pass. And if your answer was just a guess and you don't have really a logical reason for it, you can also just say that. That's fine. But let's go to Courtney. You said 0.5 micrometers. What was your reason for choosing that number?
I just kind of went with the eyeballing method and tried to count how many widths I thought I could get in there. And I counted about 10.
Mm-hmm. So you visually stood them up side by side on this little line.
Stood them up using my pinky finger.
Yeah.
I counted about 10 in there.
Yeah. And then you went 5 divided by 10 is 0.5.
Yep.
Yeah. So you and Erica are pretty much on the same page there. I agree with those. Now, this is the one I got. Is not important that this number is slightly different than the majority of people said 0.5. Some people said 0.4. Some people said other numbers. Some people said 0.7. But if that's how you did it, is just kind of using that scale bar as a gauge, then that's the right way to do it. Now, if it's 0.4, what is that in nanometers, guys? Quickly put that into the chat. Why not? Right. Don't forget your units. Never forget your units. Good job. Okay. Yeah. So we're going from micrometers to nanometers. Nanometers are smaller. There's going to be more of them. So we multiply by 1,000. 0.4 times 1,000 is 400. Okay. So when we measure microbes, it's helpful to a little bit understand like the range of the different scales we're looking at. This red line is a logarithmic scale. What that means is, unlike a linear scale where every evenly spaced region is like a predictable amount, and there's like one of these, and now this is 2 and this is 3, every evenly measured space is 10 times larger than the last one. This is an easy or a useful way to portray things that are very, very different in size or some other metric. And so what we can see is that in the millimeter range, where our eyes are working, we can see insects in the millimeter range. And then below that, we can see microbial eukaryotes and prokaryotic cells and viruses in the nanometer range. And even with the good microscope, we could see down to maybe the DNA level. So here's another opportunity for us to look at a scale bar and measure something. Giardia is a eukaryotic parasite. So it's a eukaryotic cell. Here's the 1 micrometer scale bar. What do you think? Excuse me. What do you think the height is? Pardon me. Great. Yeah. So you just look at that 1 micrometer and you go, well, how many micrometers tall is it? Just like using it like a ruler. And you're going to guess somewhere between 6 and 8 is basically what usually people get to. We can also look at the length of E. coli. Now, before we measured its width, let's look at its length again. It depends which rod you measure, so you might not get the same number as me. But here is the 1 micrometer scale bar. Use that to determine how long these E. coli rods are. Yeah, somewhere in the 1 to 3, 2 to 3 range. It depends on the rod you're measuring. Some of the newborn ones are a little on the shorty side. Some of them are longer. Here is a bacteriophage. This is one of the virus types. This is the virus that infects bacteria. We see a little scale bar here. How wide are these T bacteriophage? Yeah. So, they're pretty close to the width of the scale bar. So a lot of you said 50, some of you said 60. That's all in the right ballpark. Now, one of the things we will readdress in lab this week is the difference between these 2 key features of a microscope. When we're looking at things with a microscope, then we obviously know that microscope is capable of magnification, meaning making things bigger. But it's also really important to understand what its resolving power is, or its resolution. So magnification is defined as basically just making things look bigger, right? Apparent increase in size. On a light microscope, remember that there's ocular lenses you look through, and then there's an objective turret, right? You select one of the objectives. How do you calculate magnification on such a microscope? Put that into the chat. Right. Both sets of lenses, the ocular and the objective, have magnification. The ocular is not usually interchangeable. It's whatever it is. 10x is quite common. The objective turret has several alternatives, and you pick one, and whichever one you're using, that's got some kind of magnifying ability. To get the total magnification when the microscope is in use, you just take the ocular magnification times the objective magnification. So it might be something like 10x and 40x is 400x. Now, is that magnification the major or sole factor in determining what you can see with this microscope? Right. Obviously not, because we're not done with this slide. I would argue that resolution is at least as, if not way more important. And so what is resolution? What does good resolution provide that bad resolution doesn't do as well? Yeah, good resolution provides more detail and poor resolution provides less detail. Now, how do you calculate or quantify it? You can calculate it. You can say it's exactly this. What is that metric that we're using, or what is it called when we do the calculation? Yes. Yeah, we can calculate something called resolving power. And resolving power literally means what? If we were to do the equation, I'll show you it in a second. If you've done your background reading, you've seen it. But resolving power, there's an equation to calculate it. You get an answer with units. What is that answer telling you? Right. That's correct. The number that you get when you do this calculation tells you what's the smallest thing that this piece of equipment can visualize. We have a resolving power we've already talked about. We just didn't really say it in those words, right? We said what the limit of resolution is. So the limit of resolution is another way to say resolving power. The limit of resolution of the human eye, or its resolving power, is typically what? Put that into the chat. The resolving power of the human eye. Yeah, the limit of resolution and the resolving power, basically the same. The smallest thing that can be seen with the human eye is 200 micrometers or 0.2 millimeters. Both of those answers would be acceptable. Okay, and to prove that resolution is more important than magnification, these are two images of the same tissue. It's just the tissue is sliced very fine, and so they're adjacent. They're not exactly identical, but these two adjacent images were taken, one with a light microscope, and magnification is 450x, and one with an electron microscope, magnification 450x. So which of these images was made by a microscope that had a smaller RP number or a smaller RP value? That's right, the electron microscope on the right. This microscope has a small RP, and small in this case is better. We want a small RP because the smaller that RP number is, the smaller the things are that it can see. Okay. So the RP of our eye is 200 micrometers. A light microscope is about 1,000 times better. So what's the resolving power of a good light microscope under optimal conditions? Yeah, it's the same as saying what's the limit of resolution. The resolving power is 200 nanometers or 0.2 micrometers, 200 nanometers. So just think about that for a second and think about what we just talked about when it came to cellular life. Is a light microscope a good tool for looking at cells? Yeah, because cells are routinely larger than this, so we're going to be able to see them. Now compare that to a transmission electron microscope. Transmission electron microscope, RP is what? Yeah, 200 picometers or better. Picometers are the unit below nanometers, 1,000 times smaller. You could have just said 0.2 nanometers if you were like, I don't know what picometers are, if they're what's the next unit down, I'm not sure. But it's at least this small, it can be smaller. So can you see bacteria with a light microscope? Use your, you can either put it into the chat or you can open up that yes, no tool and reactions. It's kind of your choice. So bacteria are 0.2 to 5 micrometers, which would be 200 to 500 nanometers. And so they are definitely visible, right? They're more than the typically, I mean, maybe the width of the smallest bacteria is a little skinny, but their length is going to be fine. Bacterial flagella. So some bacteria have flagella that are about 15 nanometers wide. Are they going to be visible? Are the bacterial flagella going to be visible with a light microscope? Correct. That's more than 100 times, or more than 10 times too small. Now, why did I just say unless? What does the unless mean? Unless what? Unless we do something different? What could we do? Yes, Nicole. We could do a stain. Anybody know what kind of stain we could do? Because not just any stain, not any stain is going to work. Most stains are not going to help us. It's not going to be a gram stain, though that is an excellent guess. not a gram stain. It is a stain. When you hear this name, if you didn't know it already, you're going to just sort of smack your head. It's called a flagella stain. Yeah, we can stain the flagella. It's called a flagella stain because it's a special technique that thickens the flagella so it becomes visible. But typically you can't see it. Ribosomes are about 20 nanometers. There's no thickening method for them. So are these going to be visible with a light microscope? Correct, they're not. Now, I want you to just take that information in. What do ribosomes do? Yeah. Everybody knows, right? Everybody knows they make protein. Did you know that coming into this class? This is not new information, is it? Right. Well, how come we know that there are ribosomes if they're so small? And how long have we known that they're there? Like, how can you understand that something's there if you can't see it? Yeah, an electron microscope in the mid 1900s. So it's very recent. I mean, maybe it doesn't seem that recent to you, but it's pretty recent. Okay, yeast cells, the single-celled fungi between 5 to 10 micrometers, they can be bigger. Can you see these with a light microscope? Definitely yes. Protozoa are larger than them, so you would expect them to be visible, right? Viruses in the 20 to 100 nanometer range, can we see them with a light microscope? That's accurate. We cannot, they're too small, below the limit of resolution. Viroids are just a small piece of RNA. That's not going to be visible with a light microscope. It's too small, right? If a virus can't be seen, and a virus is a big piece of RNA usually, or maybe a piece of DNA wrapped with some proteins, a viroid is like way smaller than that. Okay, now one of the things that we're going to do in lab is use microscopes this week, and you're going to use them with oil. So it's really important to understand why oil is commonly used in microbiology when dealing with an objective microscope. So the different types of lenses that you can use on a microscope sometimes are called air objectives because there is an air gap in between the slide and the objective. And sometimes they're called oil objectives if you put oil in between and prevent there from being an air gap. So based on your reading and your viewing of the lectures, maybe your prior knowledge, what do you think about this question? A 100x air objective, so it magnifies things 100 times. So if there was like a 10x ocular, it would be 1,000 times total magnification. So 100x air objective and a 100x oil objective provide very similar resolution because the magnification is the same. Is this true or false, or yes or no? What do you think? Okay, great. So it seems to be there's a consensus. People think that even though the magnification is the same, there's something special going on that has to do with air versus oil. And so we're going to talk about that. I'm sure that most of you have talked about this before, you've seen it before. I just want to talk about it again, and we'll hear about it in lab again. But here is a schematic showing you the difference between what happens when you're trying to get information using an air gap versus an oil gap. So this has to do with a little bit of physics. It has to do with the fact that when light travels through transparent media, if it hits something different, let's say it's traveling through air and it hits glass, or it's traveling through water and it hits air. When it hits something different, it travels differently through that new medium, even though those media are both transparent. Consequently, when light travels through a transparent medium and then it hits something new, like going from air to glass, it bends. And the consequence of that for us is that the information we're getting that's providing our density of information, our resolution, depends on how many photons per square inch are coming up from the bottom light source. So let's suppose we had 100 photons coming up from this light source and they went through the slide. If there's an air gap between the slide and the objective, then as the light's coming through, some of it's bending away. Some of it bends but still is captured, but some of it's bending away. And so consequently, maybe this 100 photons becomes 50 by the time it reaches your eyes. And that means the density of information, or the amount of detail, has been cut in half. If you use oil in between the objective and the slide, as it happens, the way that light travels through glass and through oil is the same. So it doesn't bend as it leaves the glass slide and enters the oil. And so you get to keep all 100 photons, which is a higher density of information. So you get more resolution from the oil objective. So one thing of a practical nature I'll just mention is when you're in the lab, you have to know whether an objective is manufactured for use with air or oil. If it's manufactured for use with air, you cannot oil it safely. It's not going to work. First of all, you're not going to see a clear image. It's not designed for that. And also, that oil is not good for it. So on our objectives in lab, we have one objective that you're allowed to oil. That's the only one. Don't oil the other ones just because you think, oh, maybe I'll get a better resolution. Okay, so there's a simple calculation to determine resolving power. I'm sure you've seen it in your prep. But again, let's just remind ourselves what this equation is going to tell us. The smallest thing that can be seen is what it's going to tell us. What is that equation? Put that into the chat if you have not done so already. Some of you have jumped ahead and are thinking on like supersonic speed wavelengths, and you've got that equation already in the chat. Yes. The wavelength of incident light divided by 2 times the numerical aperture of the lens. The wavelength of incident light divided by 2 times the numerical aperture of the lens. I've talked about this already, you know this already, but quickly, when you calculate this number, do you want it to be small or big? That's right. You want it to be as small as you can get it. And so given that that's the case, and given that this is the way to calculate resolving power, and wavelength is on the top part of this fraction, do we want a short or a long wavelength? Correct. We want a small wavelength here because that's going to give us a smaller number over here. And when we look at this fraction, we should also note that the NA, numerical aperture, this number that's on the lens that has to do with its resolution power, the NA is on the bottom of the fraction. To make this number small, do we want NA to be big or small? Are we going to go for larger NAs or smaller NAs? Yeah, the larger the NA, the better. Short wavelength, long NA, that's the best. So here's an example. We'll revisit this in lab. By the time you get there, you'll be like, ah, this is old hat, I'm a pro. But let's suppose you had a microscope. It had a 15x ocular lens, okay? That's the one that you look through at the top that you don't change. And then on the objective turret, there were 2 options, a 60x air and a 100x oil. The 60x air has an NA of 0.75. The 100x oil has an NA of 1.25. Also, you have 2 possible light sources, an orange light source, 600 nanometers, and a cyan light source, 500 nanometers. So let's start with, what's the total magnification using the 60x air? Total magnification using the 60x air. Yeah, 900x. And how did you calculate that? You took 15 and you multiplied it by 60. Pretty straightforward. Obviously, if you take the 100x oil, the total magnification is going to be 1500x, right? As you take 100 times 15. So that's the magnification. What is the best light source that you want to use if you're trying to get a good RP? Great. So everyone says cyan. Really briefly, raise your hand and tell me why. Someone raise your hand and tell me why. Erica Marston, go ahead. Cyan is best because?
Because it's the shortest wavelength.
Correct. It's the shortest wavelength. Good job. And which of these objectives should we use? Raise your hand for that one. Which objective should we use? Raise your hand if you want to say which objective we should use. Andrew, what would you say? Which objective and why?
I'd say actually, I'm not sure.
Absolutely fine. Thank you for answering though. And if you're ever unsure, guys, just saying you're unsure is a fabulous contribution and I really appreciate it. Okay, Stephanie, you also raised your hand. Can you tell me what you think?
The NA with 1.25
So
because
this
it's
one here.
bigger. Yeah.
Bigger NA. Yeah, good. Okay, now I would like you to calculate the resolving power using, in your opinion, the best objective, which some of you have said is this one, and the best light source, which some of you have said is this one. What's the RP of that combination? Put that into the chat. And if you'd like to show your work, that would be really helpful for people who, you know, we all vary in terms of how comfortable we are with different equations. Some people find this very easy. Some people struggle a little bit. If you show your work, that's really helpful. Great. And when you're giving the answer, don't forget to use units. And what are those units going to be? Well, let's have a look at it. The NA has no units. It's a unitless empirically derived number. The wavelength has units though. Those are the units that are going to show up in the answer at the end. Great. Yes. So we take the wavelength of the light, 500 nanometers, divided by 2 times NA. This is 1.25 times 2 is 2.5. 500 divided by 2.5 is exactly as Brooke says, 200 nanometers. And many of you had that answer as well. Nanometers being units. So would this combination allow you to see the smallest bacterium, Mycoplasma genitalium, that is 250 micrometers in diameter? So quite small for a bacterium. The around that causes STDs. Yeah, it would, right? Because anything that's larger than our resolving power is going to be visible. Okay, so here is our answer. Here's our picture of our good friends, the nasty bacteria. Okay, one of the major focuses today, what we want to make sure we cover for sure is the types of microscopes and what their strengths and utilities are. Now, here are the light microscopes. These are images of the different light microscopes. We have the same organism viewed by these different microscopes. So as we go through these different letters, I'm going to ask you to name them based on the image. Okay, so what is the one shown in A? What microscope was probably used here? That's right. Transmitted light goes through the specimen. That is bright field, so that the background is bright. When the light is transmitted directly to the specimen and it hits something, that part is dark. So these dark areas are dense areas. Now this image was taken by a microscope where the light does not go directly through the specimen. It's actually, it comes in at an angle, and only if it bounces off of something does it enter the microscope lens. So the areas of light are denser and the background is what's dark. That's right. This is obviously a dark field microscope. Over here we have something that we have actually quite good contrast in the middle of the cell, unusually high compared to this one. And that has to do with the fact that both the lights collected in a way to kind of increase the contrast between the different bits inside the cell and increase our resolution, our ability to see inside of the cell. Yeah, this is a phase contrast. And last but not least, over here, we have my favorite microscope, which gives us, for a light microscope, quite good resolution, especially gives us a beautiful picture of the topography or the three-dimensional surface. That's right. That is known as differential interference contrast, and it's generated by using 2 beams of light to create a three-dimensional image. Now, which of these would be the best one to choose if you wanted to look at live samples and you did not mind having quite low resolution. You want to look at live samples wiggling around, and you really don't mind having low resolution. You're not bothered by that potential shortcoming. Right, exactly. Dark field is the best really in that it's the best way to look at live samples as long as you don't care about what's inside of the live samples. You're not going to be able to see much inside. The dark field is quite good. The general workhorse for looking at stained fixed samples. Indeed, the same microscope we're going to use. We're going to use a bright field microscope in our labs. If we need to see what's on the surface, look at some surface detail, maybe see some vacuoles. This is a technique that can allow you to see something called the contractile vacuole. In this little slippery guy, you can see it right here. It's DIC. It's useful for that. If we do need to see inside the cell, especially if we need to see inside of the cell of a live cell, we do actually need some resolution. We can't dispense with it. We could do phase contrast. Now, these are the light microscopes. There's a laundry list of other ones that we will talk about. The level of resolution that we need to see something really just depends on what we're trying to see. And again, we've talked a little bit about different parts of the cell or different things we might be trying to look at. But after the break, so I'm not trying to be confusing here. We're going to have a break. And after the break, we're going to do a breakout. But we're going to give you some information before the break so you can start thinking about it if you want to be extra prepared. But in your breakout, what I'm going to do is send you off in little teams to find out the sizes of all these things. And so in your team of about 6 people, each person within the team will be tasked with finding out the size of different components of the cell that you've heard of before. And in your team, you'll sort of report back to each other, and you'll hopefully have a decent picture of these different sizes so that when we come back together as a class, we can ask some questions. So the breakout will happen after the break itself. We are at pretty much break time right now. But I just wanted to leave this up so you know what's coming, and then I'll also leave up the time. So we're going to take 10 minutes. That means we're going to be back together at 11:05. I will see you then. All right, breakouts close in 5 seconds. We'll have everyone come back to the main room. As you're coming back in, remember to turn off your video. Excellent. Okay, welcome back, everyone. So hopefully as a group, you had a chance to look all of these up. It's not required that you memorize any of these in general, but it is very useful to have a sense of scale. And so we're going to apply that now. So based on what you learned in your group, you're going to vote with your yes, no tool. So go find that now if you can in the reactions or participants menu, that yes, no, that green check mark and that little red X, go find it. If you can't find it, you can use the chat to put your answers in, but it'll be much more efficient if we use this tool. So I'm going to display each of these items, and based on what you learned in the breakout in terms of size, you're going to tell me if you think that it is visible. Can you see with a light microscope the nucleus of the cell? Use your yes, no tool to answer that if you can. Okay. And so yes, a green check mark if you think yes, and a red X if you think no. Perfect. Most people think yes, that is true. Why? Probably what you found was that your nucleus is about 5 to 6 micrometers. So that's definitely much bigger than the limit of resolution of a typical light microscope. The other thing that you might want to consider is that this is sort of in the range of a larger prokaryotic cell. So nuclei are only found in eukaryotic cells, and their size is kind of like a prokaryotic cell. You kind of need to be a eukaryotic cell to even be big enough to have a nucleus. Mitochondria. Based on what you found out about mitochondria, yes, visible with a light microscope. No, not visible with a light microscope. Just based on its size, the answer would still be yes, although it is true you'd need special dyes because mitochondria are transparent like everything else. But if you have the right dyes, it would be big enough. It's about a micrometer, which again is about a typical size for a prokaryotic cell. It's a structure only found in eukaryotes. So if you were trying to answer the question like, does a bacterium have mitochondria? Just remember that the mitochondria is the size of the bacterium. It'd be kind of like asking if you happen to have any elephants inside of you. Now, lysosomes, what did you find out about those? Big enough to see yes or no? Okay, 50-50 on this one. That's kind of appropriate depending on where you found your information because they actually vary in size from 0.1 up to 1 micrometer. And if they're on the smaller side, the answer would be no, because 0.2 is the limit of resolution. And if they're on the larger side, the answer would be yes, as long as the dye situation was right, because, you know, but based on their size, sometimes the answer would be yes. All right, width of a microtubule, I asked if this would be visible with a light microscope. What do you think, yes or no, based on what you learned? More of you are saying no, that's accurate. The width of a microtubule is 25 nanometers. That's almost 10 times below the limit of resolution. Ribosomes, any ideas? We talked about this earlier. You may not have recalled it. That's fine. You don't have to memorize it, but based on what you talked about in your group, most people are saying no, that's accurate. They're about the size of a microtubule, slightly smaller actually. Width of actin, visible, yes or no? Yeah, hard no. Everyone's like hard pass on that. The most confident answer so far is actin is not visible. It is only 7 to 8 nanometers wide. So what can you use instead if you can't see these things with a light microscope? How can we know they exist? Put that into the chat. And just for those of you who are interested, Hannah has reposted the Discord link in the chat right above the section where everyone is saying electron microscope, which is the right tool for the job. Really, it's not so much, the point of this isn't really like to have you decide in the lab what you're going to use, because you only get access to a light microscope. It's more to help us appreciate what electron microscopy did for the field, which was revolutionary. So the transmission electron microscope is a very powerful tool. It has definite pros. It also has some definite cons. So if you can tell me what would be one advantage of using an electron microscope, raise your hand. One advantage of an electron microscope. Brooke, go ahead.
It has very good resolution.
Excellent resolution. We can see things that are much, much smaller than cells. So that's fabulous when we're trying to learn about cells. Okay, so it has more than 1,000 times better resolution. This is like the bare minimum better is 1,000 times, and it could be 10,000 or 100,000 times better. It allows you to see the interior world of the cell in great detail, which is amazing. Any cons? Raise your hand if you know of any reasons why. What are the limitations or the shortcomings or things that just like it can't do? Andrew, go ahead.
It kills whatever it is looking at. It'll kill it because of the electrons.
Yeah. So I'm going to rephrase that
And it
a
has
little
to be
bit.
sliced super thinly.
Yes. Yeah. It has to be super, super thin slices. So that requires a special tool. And it's not that the electron stream kills the sample. It's more that in order to see it with electrons, we have to kill the sample. And it's not even that necessary that the sample is dead per se. It's just that to prepare it to be visualized with electrons, what we have to do, slice it very thin, as Andrew mentioned. So that's not going to be super awesome. Like so thin, like you're slicing a cell into many pieces. That's how thin. But also, you have to treat the sample usually with something like heavy metals, because electrons are so high energy that they would just zoom right on through the tissue without even noticing it was there. So a common way to treat the sample is to soak it with heavy metals. And then the heavy metals kind of bind to all the dense areas. And when the electrons go through the sample, they hit the heavy metals and they bounce off. And it's actually us capturing the bouncing off of the electrons that tells us what we're seeing. So it's not as easy as like looking through an ocular. You can't just look at it with your eyes. You have to send that information. It goes to a film and usually nowadays to a computer, and then that image is generated for us. So the sample has to be prepared in such a way that it is not alive. It has to be fixed with heavy metals. We have to use a vacuum in order to visualize it. And the whole procedure and the whole like equipment itself is very big. You can't carry it around the way you can carry a light microscope. It takes up a whole room. You need a lot of training to use it, and it's expensive, but so worth it. And when this technology was developed, it was amazing. So here's what you can see for 200 years or 400 years with a light microscope. This is an streptococcus, basically. And this is what you can see when you use an electron microscope to visualize the same cell type. We can see all kinds of things. Just for fun, I'm going to cold call you to guess what some of the items are that are pointed to, and then I will tell you why I'm doing this. I always have a reason. But first I'm going to cold call you to guess. And again, you don't have to be right. You don't have to know. You don't have to guess. You could say pass. That's fine. But if you want to take a guess or you think you know, go ahead and say what it is. We're going to start with Sarah for A.
I'm not sure, but I think it's Syria.
Celia? Okay.
Yeah.
Let's go to Mai. What do you think B is?
I was going to say B was Cilia.
Okay, great. Lanisa, what is C?
I am going to pass. I've been wondering. I don't know.
Yeah, that's a great answer. I don't know is a great answer. Looking at this for the first time, that would be a really common answer. What the heck is that? What about D, Melissa?
I think I'm gonna pass. I'm not quite sure.
Yeah. And then for E, I'm going to ask Aminata. What do you think? It's pointing to some kind of something in the interior of the cell. Aminata, what do you think E is pointing to?
I think I'm gonna pass too. I'm not really sure.
Yeah. All right. Now, before I reveal the answers and talk about why we're doing this, does anyone have a guess that they want to share that hasn't been said or about any of these? You can just raise your hand and say, I think A is, or I think E is, or whatever. Lanisa, what would you say you're seeing here that you think?
I was thinking maybe A or D were cell membranes.
Okay.
And that's kind of all I had.
Awesome. And Mai, you had your hand up as well. What would you say?
I was going to guess that C was the Golgi apparatus.
Great. Anyone else want to take some guesses? Notice I'm not commenting on these guesses. I think these are all great. I think I don't know is great. Andrew, go ahead.
I would say C might be a nucleoid.
Okay, C might be the nucleoid. Mariceli, what's your guess about any of these?
I was gonna say the C is the nucleus and maybe E is the cytoplasm.
Great. And Haley, what's your guess?
Oh, I was going to say E might be some ribosomes, just to take a guess.
Great. Okay. Awesome guessing, guys. Guess what? Why did we do this? You don't have to look at a picture like this and tell me on an exam what all the things are. That's not the point. The point is that the first time this technology was used to look at cells, it was clear that there was a lot of interesting stuff going on that could not be seen with a light microscope. And pretty much we didn't know what any of it was. I can tell you now what it is, okay? But it's not obvious until you know what it is, right? So let's start with A. A is pointing at the sort of border here. This is a bacterium. Okay, this is a bacterium, which means it's a prokaryotic cell. It's a bacterium and it has a cell wall, right? And now we know that probably that cell wall has chemicals in it. What is one of the key chemical components if we did a chemical analysis on the cell that we would find that's part of the structure pointed to an A? Put that into the chat if you know what it is. Yeah, peptidoglycan. And guess what? If you knew that, you knew more than the really intelligent people who were looking at this for the first time. They were looking at the first picture no one else in the world had seen, and they were like, I don't know what that is. B is, because this is a so cilia is a good guess. Because this is a prokaryotic cell, as we'll talk about next week, they don't have cilia. Cilia are actually quite large structures. They do look like this if you were looking at the right magnification. But because this is a prokaryotic cell, we can be confident this is like fimbriae. Sometimes they're called pili, but they do kind of have a cilia-esque appearance under the right magnification. D here is pointing to the cell membrane. And it's amazing that, it's not really easy to see, but an electron microscope will kind of allow you to see where it is. C is the nucleoid. It's this region where the chromatin is kind of just piled up inside the cell. And E is ribosomes, the little dark spots. Okay. Now let's have a look at some cells maybe you care more about. These are blood cells. These are lymphocytes. Now on the left is a blood smear. What are all these little pink circular guys in this field of view? Pink circular guys. What are all these guys? Yeah. Yeah, this is a blood smear. These are red blood cells. So what about these kind of larger purpley pinky guys? Yeah, white blood cells, lymphocytes. Okay, so white blood cells, which include lymphocytes, I should say. This is an image that these are actually monocytes here, and these little smaller guys are lymphocytes. But these white blood cells known as lymphocytes, they come, you may recall, from ANP in two flavors, T cells and B cells. And on this light microscope, you cannot tell the difference between a T and a B, can you? Just like if I told you this is a cluster and two of them are T's and twos are B's, you might be like, the smaller ones could be something, I don't really know. You can't tell the difference. But if you look at these cells under an electron microscope, suddenly you can tell that there's two distinct types. So first of all, based on just what you're looking at and what you know about T's and B's, what is it that you know about B cells? What is their major job? What do B cells do? And these are activated cells, which is to say they're not the naive ones that are just born. They've sort of started doing their jobs. What is it that B cells do for a living? What's their job? That's right, Courtney. That's right, Haley. Yeah, Courtney's right. Haley, what you've described is an excellent description of what T cells do. Courtney said make antibodies. Haley said cellular immunity. So that's the two different, yeah, it's easy to get them mixed up, right? That's the two different jobs. Like T's provide cellular immunity. We'll talk more about what that really means at the end of the quarter. And B's make antibodies. We'll talk more about those details at the end of the quarter also. But right now we can just admire how beautiful and different they are. Which one's which? What do you think? So let's say the image on the right that looks like a fingerprint in the cytoplasm. Which one is this? Is this the T or the B? Put that into the chat. Just put your guess in. And this is one where you're not allowed to abstain. I don't expect you to know, but just put your guess in as to which one this one is on the right-hand side. Fabulous. We have a pretty 50-50 split, which just makes sense because you don't really have much to go on. So a 50-50 split is actually kind of what you'd expect if you're kind of like, I have to guess. Let's talk about what this stuff inside could be, because it's clearly different, right? A major difference between these cell types is how much all this googly wiggly stuff inside the cytoplasm is inside of this cell versus that one. What do you think this stuff is? All of these like squiggly membranes, what's that? Yeah. So Golgi is not a bad guess. Some of it is Golgi. Some of it is smooth ER. Some of it is ribosomes. They're all kind of connected in a system. The major component of which is the endoplasmic reticulum. Remember that? That kind of looks like this. The endoplasmic reticulum is part of a system that generates proteins, but doesn't generate all the proteins in the cell. It just generates the ones that are going to go to the membrane or that are going to leave the cell. Does that give you a hint as to which cell type this might be? And we have a lot of it. It's like we have a factory for making proteins. They're going to leave the cell. So what cell do we think this is now based on that? Yeah, that's right. Now everybody's kind of more on the side of the B cell. Let's think about what T cells do, right? If this is the B cell, what's different about this guy besides not having as much of this endomembrane system? What's different over here that we can see that's kind of not so much present here? Yeah, Nicole. Yeah, the shape of it isn't perfectly smooth. It's got all these little blebs. Blemises as fingers. Yeah, that's a good description. That's really what they are. Reaching out to kind of feel stuff, to provide cell-to-cell contact for cellular-based immunity. Okay, again, I'm not trying to get you to look at these images on a test and tell me which one's which. It's not, the point is not to memorize stuff the way you would in A&P. The point is to understand what kinds of information is provided in this field and how we can use that logic to figure stuff out. Okay, now I'm not going to cold call anyone for these, but if you see stuff in here besides what we've talked about, which is the endoplasmic reticulum, but if you see anything in here and you're like, I think I know what that is, I'd love for you to raise your hand and just tell us what you think it is. And I'm just going to first lower the hands that I see because they might be left over. So now go ahead and raise your hand if there's one of these you want to take a stab at. Mariceli, go ahead. What's one of these letters that you think you know what it is?
E, it could be lysosome.
E could be a lysosome. Okay, great. Anyone else want to take a guess at what some of these things are? I mean, this is for 0 points, just for fun. Nicole, go ahead.
Maybe A is the nuclear membrane.
Yeah, A could be the nuclear membrane. Good job. Okay, anyone else? Mariceli, did you have another one?
Mitochondria for B.
B could be mitochondria. Yep. Anyone else have another guess? We got, so far we've got A, E, and B. And we've got D and C. We already talked about C and we talked about D. So never mind, y'all have covered everything. And that's, yeah, those would be great guesses for all of those things. But again, I just want to remind you, first time really smart people were looking at those images. They were like, I don't know what that is. I think we're going to call it a Golgi. Let's call it a Golgi. Great question, Andrew. Let me go back to answer that question. Would D be Fimbriae? So Fimbriae is a prokaryotic structure only. So it wouldn't be. You would find it on bacteria, but not on the, it's so tiny, be much smaller than this. But you wouldn't know that unless you knew, oh, this is a eukaryotic cell. It could be cilia. It's unlikely, only, again, and just as experience, it's a good guess. I love that. But cilia are actually skinnier and they're much more regular. See how these are all kind of different widths and kind of squiggly? Cilia are very uniform in their width and they're longer than this. They're not as long as flagella. And you'll see them kind of like hairs. They look more like hairs than like little fingers. But those are great guesses. Love it. Okay, now there were some other microscopes you needed to know about. So after bright field, dark field, phase contrast, DIC, and transmission electron microscopy, what else did you have to know about? Put into the chat the name of any of the other microscopes you have to know about from this section. Yep. Atomic force, scanning acoustic, confocal, also known as a laser scanning, fluorescent, yes, also known as UV. Good job. Yeah. So here's the 4 other types. Now I'm going to give you a few minutes to remind yourself what is used for illumination and anything else you might remember, like what's the most common or important utility of this microscope, what might be some of its limitations. So just take a minute. It's not going to be 4 minutes, but it's going to be more like 2 minutes to think about what those are, if you can remember, and I'll cold call some people and then we'll chat about it a little more. All right, so I'm just going to cold call a couple people. Give me anything you know about each of the microscope when I ask you. I'll ask you for a specific microscope, and then we'll kind of open it up for people to add anything that they know that hasn't been mentioned. But I'm going to start with Alyssa for the UV, also known as the fluorescence microscope. What do you remember about its source of illumination, things that it's particularly good at, any limitations? Are you able to unmute, Alyssa? And remembering always that passing is a total option.
I would say that it's I think I'm gonna pass. I'm sorry.
That's absolutely fine. And thank you for being the first brave one to do that for this activity. Everyone else is now breathing a sigh of relief. They're like, thank goodness I didn't have to be the person who had to pass. I will ask one other person. Let's start with Braden.
I don't really remember much about all these microscopes.
All good. Okay. I'm going to ask one more person and then we'll open it up to everyone to just raise your hand, because some people I know are just like, I know something. Let's go to Erica Lowry. Do you remember anything about the UV microscope?
That it does have great resolution.
It has better resolution than the light microscope. Good.
Yeah.
Anyone else want to add anything? Go ahead and raise your hand if there's something about the microscope that hasn't been mentioned yet. Jordan, go ahead.
That it has high contrast against dark background, and then it can observe small structures inside the cells.
Yeah, the contrast is really an important part of it. And it has definitely better resolution, as has been mentioned, and it allows us to see subcellular detail. It's great. Nicole, what would you like to add?
So you can see things that are alive, but you can't do it for too long because it damages the tissue. And a pro of it is that you can see fluorescent things.
Yes. So the source of illumination is in the UV spectrum, which allows us, and it also, that UV light allows us to see fluorescent things. There's several proteins and dyes that when you put a UV light on them, they reflect back visible light. That's really useful. So that's why it's called a fluorescent or UV microscope. Because UV, ultraviolet light, has a shorter wavelength, the resolution is better than a light microscope. The contrast is better. It allows us to see subcellular detail, allows us to actually co-localize things. And as was mentioned also, while you can look at live things, unlike the electron microscope, you can't look at them for too long because UV is damaging. Okay, let's move on to the confocal. For this one, I'm going to initially ask, let's see, Alina Park for the confocal. What do you know, if anything? If you have to pass, that's just fine.
Can you hear me?
I can hear you.
Oh, okay. I don't really know, but I would say it has a high resolution.
Yes, very high resolution for something that's not an electron microscope. High resolution, yes, excellent. Let's ask John if you have anything to add to that for the confocal microscope.
No, sorry, I'll have to pass.
No worries. Thank you, John. And last person I'll ask before I open this up is Olena. Olena, do you have anything to add to the confocal microscope?
Yes, it can reconstruct 3D pictures, so we can see like the cells of 3D pictures very well.
Yeah.
To observe it.
That's a very important feature. Excellent. Now, before I say anything about it, does anyone have anything they want to add? Haley Kronberger, go ahead.
Can you hear me?
Yep.
Okay, I remember from the lecture that it was cool because it can illuminate each plane of a specimen. It's like really precisely.
Yes, and that's exactly how that high resolution is derived, is by doing optical sectioning and then reconstructing it with a computer. That's an amazing feature. Courtney also had her hand up. What would you like to add, Courtney?
I was just going to say that it uses a laser and that it's great for depth perception.
Yeah, it uses a laser as its source of illumination, and the laser can often be in the UV wavelength. You can really decide whatever you want it to be, but it's usually in the UV wavelength, but it's actually more precise. We can get into lots more fun things about that. Andrew, what would you like to add?
I was going to say the laser as well, but also it has it's good for subcellular imaging.
Yes, exactly.
And then also, and then can provide a live time lapse.
Yes, that's right. Live imaging is better than UV because the laser is more targeted, so it takes longer to cause damage. And time lapsing is a common way we use that for that reason. We can go for over time and look at something alive.
Is
Nicole, go ahead.
Oh, sorry. I was just going to say, is it dangerous using a laser?
Yeah, you have to be careful. Yeah, because
Okay.
lasers are damaging. You don't want to shine them in your eyes. It's done with a lot of care to where the laser is pointing.
Okay.
Yeah, great question. Excellent. Okay. Next up, we have the scanning acoustic. Raise your hand if you know what the source of illumination is. Actually, just put that into the chat. Most people, I think, can guess. Scanning acoustic, what's the source of illumination? Yeah. That's right. It's sound waves. It's like a miniature sonar. Very useful for looking at things like in place, like a microfilm or a biofilm that we don't want to disturb. We can get a nice three-dimensional image of it without having to lift it off of its surface. It doesn't give us very good cell-to-cell resolution, certainly not subcellular. And then atomic force as its illumination, what does it use, guys? We've got sight, we've got sound. What's left for the atomic force? Yeah. It's basically like a little touch probe, diamond probe, a metal diamond probe, and it's feeling along the surface of things. Typically used to visualize what? What do we use these for? Surfaces, that's right, especially membrane surfaces. It doesn't go very deep, so if you have something that has depth, it's going to be a bit of a problem. Okay, so here is a summary, and this is just to kind of remind you, like, what do you need to know about these microscopes for this section? We don't have time for this activity. This would be really fun to do, but I might actually start with this on Wednesday because we have time. So what I'd be asking then is sending you to breakouts to talk about which of these microscopes you'd use in each circumstance. So we might do that breakout next week if we don't, or next class. If we don't, you can do it on your own. Okay, so that's naturally where we would break for the class for next week. What I want to make sure I talk about is, let's see. Yeah, I just want to make a quick comment about the unknown ID project. And that is that you have selected your unknown ID. Good job. Now it's time to start part one. So read about the project, read about the part one instructions. Basically, you're going to be sending an email to the technician asking for the gram stain for your unknown, but there's a particular wording that I want you to use. So I want you to put your ID number and name into the subject of your email and send it to microlabs@everettcc.edu and request the gram stain. And then you're going to reiterate, I'm so-and-so, my ID number is such and such, and I need the gram stain. The one thing that I want to say now, and I'll try to remember to say it later when it's more relevant, you're going to be sending a lot of emails to this email address. It would be best if when you make a request that you start a new email chain instead of just replying to her last reply to you. The reason for that is it's way easier then for us to discover if we've dropped the ball on something, like is there something that hasn't been done yet? So please start a new email chain every time you have a new request. Yes, Andrew, most of that's in the information pages on Canvas. The only thing is this nuance of starting a new email chain is not on there, but the email address and the instructions for how to request it are all there. great question. Okay, so next class, we're going to talk about staining, and we may start with this breakout. I don't know, depending on timing, I'll think about it. And there's also a Kahoot we'll do next time, which is fun. But in the meantime, if you don't have any questions, then by all means, go ahead and enjoy the rest of your Monday. If you do have questions, stick around, raise your hand, and I will do my best to answer them.